Hello, everyone. Today we have a lesson on Module 5, which is cellular energy. And there are two main topics in this module, photosynthesis and cellular respiration. So rather than going through the book like I did last time, we're going to watch a video and pause it and take notes together as we go, so that maybe you'll have a better understanding of these processes when we finish. So go ahead and get some paper, a pencil, label it Module 5, Cellular Energy, and I will share my screen and we'll get started with the video. Okay.
I've daydreamed before about what it'd be like to have a special ability that other organisms can do. For example, to be able to fly like a peregrine falcon, or to be able to walk up walls without any gear, like a Texas bandit gecko. But if I told you I really wish I could have a special ability of a plant, you'd probably be confused. What can a plant do that's so amazing? There's a lot of cool things about plants, actually, but in particular, I really wish I could do photosynthesis. And it's not just plants that can do this process. Some protists and some bacteria can too, for example.
Okay, go ahead and write that at the top of your paper. This is our first topic, which is photosynthesis. So I'm taking notes too, and I wrote photosynthesis at the top.
But plants will be our main focus for this video clip. Animals and amoebas have missed out on this ability, but we benefit from it greatly, as this process also produces oxygen, a gas that we need. Plants in general are major producers, making them indispensable in food webs. Many of our medications and foods come from plants. We need plants. So understanding the nature of the process that plants use to make their own food is paramount. So when I say make their own food, I'm talking about making the sugar that they need, specifically glucose. You need glucose too, but you get it from what you eat. Plants, however, get to make their own glucose in photosynthesis. Here is the balanced overall equation for photosynthesis, similar to what you'll find in many introductory biology textbooks. As you will notice, it has some similarities to aerobic cellular respiration. Recall that cellular respiration is used to make ATP, which is an energy currency, and it's done by plants as well as animals and a lot of other organisms too. You can see how these reactants, inputs of photosynthesis, are included in the products, outputs in cellular respiration, and the products, outputs of photosynthesis are included in the reactants, inputs of cellular respiration.
Okay, so these are the two processes that we're studying during this module. So go ahead and let's write down the photosynthesis reaction. So we're going to write down 6 CO2 plus 6 H2O produces C6H12O6 plus 6 O2. And we're going to label it that this is our glucose. The rest you should know. So we have our 6 carbon dioxide molecules reacting with 6 water molecules, introduction of light, which I'll put above my arrow here, of light to produce 1 glucose molecule, C6H12O6, plus 6 oxygen molecules. And that is the chemical equation for photosynthesis.
Well, this doesn't mean that they're simply reversed. It is interesting to see what they have in common. So while both plants and animals need glucose for cellular respiration, plants don't have to be in search of glucose because they make it. Plants have adaptations to carry out photosynthesis in a variety of environments. One thing plants have to do is capture light. Plants can use light-capturing molecules called pigments. Recall that visible light has different wavelengths, and those different wavelengths of light have different colors. If you've ever played with a prism before, you can see how light can be separated into a rainbow of colors due to different wavelengths. So a pigment that plants commonly use to capture light is called chlorophyll. Chlorophyll does a great job at absorbing red and blue light, but not so much green light. Chlorophyll.
Okay, so let's go ahead and write down chlorophyll. It absorbs red and blue light, reflects green. Okay, so now you'll have more notes that chlorophyll absorbs red and blue wavelengths of light, reflects green wavelengths of light.
Reflects green light, resulting in many plants appearing green to our eyes. There are pigments besides chlorophyll that work with different wavelengths of light, and this can explain why green is not the only color you see in plants. Chlorophyll is a pigment that can be found in the chloroplast of plant cells.
Okay, so we're going to put an arrow here and put that chlorophyll is found in the chloroplast. So it is found in the chloroplast in plant cells. Okay.
There are two major processes that occur in the chloroplast that together make up photosynthesis. They are the light-dependent reactions and the light-independent reactions. The light-independent reactions can also be called the Kelvin cycle, or even, less commonly, the dark reaction. Sounds intriguing. We're going to talk about both of these briefly, and please remember, like most of our videos, this is pretty general. We've got some further reading links in the video.
Okay, so we have two different reactions happening in the chloroplast. We have the light-dependent reactions and the light-independent reactions, which is also known as the Kelvin cycle. So I just drew two arrows to show that we have the light-dependent reaction and the light-independent reaction, also known as the Kelvin cycle.
Description where you can explore a lot more detail. So, light-dependent reactions happen in the thylakoids, little compartments in the chloroplast that contain pigment.
Okay, so we have the thylakoid. I'm going to erase. Bryson, will you hand me a paper towel, please, to erase quickly? It doesn't have to be wet, just quickly. Okay, so I'm going to go ahead and write, draw this. I'm going to draw this chloroplast, and then I'm going to draw the thylakoids stacked inside, which make granum, which are the little stacks. So I'm going to draw this is the chloroplast. Then I'll draw my stacks of discs, which are my thylakoid. And then a whole stack is called a granum. So I drew the chloroplast, and I have my thylakoids labeled, just one of them, and then a whole stack labeled as well.
A collective stack would be a granum. Multiple stacks would be grana. In the light-dependent reactions, light is captured, and water, which is a reactant in the photosynthesis equation, is split. That means if you think of the chemical formula for water, which is H2O, it is split, so you get electrons, protons, and oxygen. So oxygen is also a product of the light-dependent reactions. The light-dependent reactions also produce ATP and NADPH, which we'll get to in a little bit. Both the ATP and the NADPH will be needed for the next process.
Okay, let's go ahead and get this into our notes. So we're going to draw a granum, which is a stack of thylakoids that are located in the chloroplast. We're going to input light as well as the breakage of water here on that side. And I'll just put an X here to indicate that it's being cut. And then we're going to have an output of ATP and NADPH. And we're going to learn what ATP is in a little bit, and NADPH. But for now, just know that this is where the light reaction occurs. Here comes the light. The water is chopped. So then you just have the protons and electrons and oxygen coming off of that to produce ATP and NADPH.
The light-independent reactions, also known as the Calvin cycle or dark reaction. The name is a bit misleading. While yes, the process isn't directly capturing light, it doesn't require darkness either. And again, it will need items from the light-dependent reactions, like the ATP and NADPH. The light-independent reactions still happen in the chloroplast, but specifically, the light-independent reactions happen in the stroma.
Okay, let's go ahead and get that labeled. So you're just going to label anywhere where there's like blank space. So I'm going to draw my arrow right here, and that is the stroma. So I just drew an arrow to the blank space between my granum.
The stroma is a fluid outside of the thylakoids. In the light-independent reactions, or Calvin cycle, carbon dioxide enters. It is taken in through pores that are often, but not always, on the bottom of leaves, and those pores are called stomata. Plants have the ability to open and close their stomata. The carbon dioxide gas enters the stomata and will be fixed by
Okay, so I'm going to draw on my notes like double arrows here because I ran out of room. And then I'm going to draw big double arrows here. And I'm going to write the Calvin cycle here. And so I'm saying that the ATP and the NADPH are going to be used in the Calvin cycle. Okay. Now I'm going to draw and define stomata. So if I have a leaf here, on the bottom of the leaf, so I drew a leaf, on the bottom of the leaf are the stomata. So if I drew a thing of plant cells right here, and I would define it as openings on the underside of the cell, or pores. Okay. Singular is stoma, plural is stomata. So I guess I should draw more than one here. Okay, stomata. And these allow CO2 to enter. So they allow CO2 to enter through the stomata on the bottom side of the leaf.
On the bottom of leaves, and those pores are called stomata. Plants have the ability to open and close their stomata. The carbon dioxide gas enters the stomata and will be fixed. By fixed, I mean that with the additional help of a major enzyme, the inorganic carbon dioxide is changed to a more usable form. The ATP from the light-dependent reactions will act as an energy currency for the Calvin cycle. The NADPH that had come from the light-dependent reactions will supply reducing power. By that, I mean that it helps add high-energy electrons to this process. So, in a very complex series of pathways, the fixed carbon dioxide, ATP, and NADPH are used to make a product that ultimately can be converted into glucose, a sugar. Whew. So let's take a look at this equation. Last time, I promise. So we have here the circled items. These were from the light-dependent reactions. Now, notice these other items, the carbon dioxide on the reactant side and the glucose on the product side. Those were from the Calvin cycle. Remember, there is so much more detail to explore in this amazing process. You can learn about the photosystems that are in light-dependent reactions.
Okay, let's stop there and be sure that we understand the last part of this process. So you have the ATP and the NADPH that came from the light dependent reaction, entering the Calvin cycle. You have CO2 that's been fixed with an enzyme into a usable form for the leaf and the plant cells, entering the Calvin cycle from the stomata. And all of those three things are producing the glucose, which is your sugar. I'll write that under there. It's a simple sugar. Okay.
Or detail of all the steps in the Calvin cycle and how ATP and NADPH will be converted to ADP and NADP+, which can then be used again by the light-dependent reactions. But before we end our short video, we do want to mention that plants have some amazing adaptations that can help them perform photosynthesis efficiently in very different environments. Many of these adaptations can involve the diversity of leaf shapes, coverings, and pigments. This is definitely worthy of a completely separate video topic, but just to give a neat example of an adaptation involving photosynthesis, consider the cactus. Cacti have a potential problem. They often live in a hot desert, and so if they open their stomata during the hot day to get their carbon dioxide, they can easily lose more water than would be ideal. The precious water can escape through the stomata if the stomata are open, and that will happen at a faster rate in the hot desert sun. But cacti, and some other plants too, can do something called CAM photosynthesis. In CAM photosynthesis, plants can open their stomata at night when it's not so hot, and they can capture carbon dioxide and chemically store it. They can then use this carbon dioxide the next day when the sun is shining and yet have their stomata closed. This allows them to avoid having to open their stomata in the heat of the day. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay. All right, the next one, so that is photosynthesis. And I told you we were doing two processes. Well, we'll have to do fermentation too, but the next one that we're going to study, so that was photosynthesis. So the next one that we're going to study is cellular respiration. So let me get...
Small business owners, use Melio to send and receive ACH payments. Are you a morning person? One of us is, and one of us is definitely not. Mainly because when I wake up in the morning, it just takes a while for me to get my energy back. It takes a lot of time and coffee for that to happen for me. Cells really don't have that luxury. They are busy performing cell processes all the time, active transport of many substances needed for their survival, for example. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's
okay. Let's go ahead and...
Specifically, is ATP. ATP stands for adenosine.
Okay, so the first thing you want to write is cellular respiration. And then we're going to write the word ATP, which is the energy currency. And the full name is adenosine meaning 3, phosphate. Adenosine triphosphate.
Triphosphate. It's a type of nucleic acid.
Okay, we learned about nucleic acids back when we talked about biomolecules. So we're going to write a little reminder here that our biomolecules were carbohydrates, lipids, proteins, and nucleic acids. And I'm going to put a big star by nucleic acids. And this is what this is, ATP. It is a nucleic acid. So I just wrote a little reminder of the biomolecules, the macromolecules we learned about when we did our paper clip and pipe cleaner activity. We learned about carbohydrates, we learned about lipids, proteins, and nucleic acids, and ATP is a nucleic acid.
Actually, and it is action-packed with 3-phosphate.
Okay, now we're going to draw ATP. So we're going to draw the base, which is adenine. Then we're going to draw the sugar, which is ribose. And then we're going to draw our 3 phosphates. Okay, so I have drawn ATP. Here is my base, adenine, sugar, ribose, and the 3 phosphates.
We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are, prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different, however, depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration, but this video is specifically going to go into aerobic cellular respiration in eukaryotic cells.
Okay, so we are doing aerobic cellular respiration in eukaryotic cells. Okay, in eukaryote cells.
That means this video is talking about the process within cells that have membrane-bound organelles, such as a nucleus and mitochondria. Eukaryotic cells include the cells you'd find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells, are going to be kind of a big deal in this aerobic cellular respiration.
Okay, let's go ahead and write down mitochondria. And it is the location where the ATP is produced.
Some of the process occurs in the mitochondria. So let's
process to make ATP occurs in the mitochondria.
Let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow.
Okay, so let's go ahead and write down cellular respiration, chemical reaction. So we have C6H12O6, which is glucose, reacting with 6 oxygen molecules to produce 6 carbon dioxide molecules, 6 water molecules, plus ATP, which is what we are looking to make in cellular respiration. And I wrote before that, I wrote that the goal of cellular respiration is to make ATP. And there's the chemical reaction.
The arrow. This equation, by the way, looks remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. Well, that doesn't mean they're simply opposites. It does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is a product. But in cellular respiration, organisms break the glucose down to make a...
Okay, so we're just going to make a little side note here. I'm going to put mine in a box, and I'm just going to write down that photosynthesis makes glucose, and that cellular respiration breaks glucose to make ATP. Okay, so I'm gonna put mine in a box here, so I know it's a little side note. So as a little side note, I have photosynthesis makes glucose, and that cellular respiration breaks glucose to form or make ATP.
ATP. Fun fact, you know how when a bean seed first germinates in the ground, it isn't able to do photosynthesis yet? Yeah, the germinating bean is relying on glucose that it has stored, and it's doing cellular respiration to break it down to make ATP so it can grow. Of course, once it starts to mature and develop leaves, it can do photosynthesis. It will be able to do both photosynthesis and cellular respiration. Plants make glucose in photosynthesis, and they break it down in cellular respiration. They can do both. But if you aren't photosynthetic, such as a human or an amoeba, you have to find a food source to get your glucose. You need glucose to get this process started, and we're going to assume that we're starting with one glucose molecule. Step 1, glycolysis. This step takes place in the cytoplasm, and this step does not require oxygen. It's considered
Okay, let's go ahead and write down that model on our paper. And we'll go ahead and put glycolysis in that. And we'll leave the other two blank for later in the video. So we'll go ahead and have this drawn. And I'm going to write glycolysis here. And I'm going to make some notes that it happens inside the cytoplasm of the cell, and that it is anaerobic
In glycolysis, glucose, the sugar from the equation, is converted into a more usable form called pyruvate. Glycolysis usually takes a little ATP itself to start up. The net yield from this step is approximately 2 pyruvate and 2 ATP molecules, and 2 NADH. What is NADH? NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now, an intermediate step occurs.
Okay, let's go back.
2 pyruvate and 2 ATP molecules, and 2 NADH.
Okay, so we need to write that down. We have glucose breaking down to form 2 pyruvate plus 2 ATP plus 2 NADH. And now I need to say what NADH is. So we have glucose reacting to form 2 pyruvate, 2 ATP, and 2 NADH.
What is NADH? NADH is a coenzyme.
It's a coenzyme that can transfer electrons. It's a coenzyme that can transfer electrons. Okay.
And it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now, an intermediate step occurs. The 2 pyruvate are transported by active transport into the mitochondria, specifically the mitochondrial matrix. In the mitochondria, pyruvate is oxidized. The 2 pyruvate are converted to 2-acetyl-CoA, which will be used by the next step. Carbon dioxide is released and 2 NADH are produced. Step 2, the Krebs cycle, also called the citric acid cycle.
So we're going to write that one here. Krebs, citric acid cycle. Okay, so we have that there.
Still in the mitochondrial matrix. The citric acid cycle is considered an aerobic process. While the cycle doesn't directly consume oxygen,
So I'm going to make notes about it. I'm going to write Krebs cycle. And I'm going to start my notes. It's aerobic, meaning it uses oxygen. Anaerobic means it does not use oxygen.
Some of the events in the cycle need oxygen to continue. To learn more about this intricate cycle where the 2-acetyl-CoA will enter, check out the further reading suggestions in the video details. Carbon dioxide is released. We also produce 2 ATP, 6 NADH, and 2 FADH.
Okay, so for this one, we start with 2 acetyl-CoA to produce 2 ATP, 6 NADH, and 2 FADH2. And I'm going to put here that CO2 is released. Okay, so now I have drawn that our 2-acetyl-CoA produce 2 ATP, 6 NADH, and 2 FADH2s.
FADH is also a coenzyme, like NADH, and it will also assist in transferring electrons.
Okay, so we need to define it. FADH2 is a coenzyme that transfers electrons. Okay, so I wrote down FADH2, coenzyme transfers electrons.
To make even more ATP. Step 3, the electron transport chain and chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is in transferring electrons to make even more ATP. Step 3, the electron.
Okay, so our last step is the electron transport chain. So now we have the electron transport chain.
Transport chain and chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is still in the mitochondria, and to be more specific, it's involving the inner mitochondrial membrane. We do require oxygen for this aerobic step. This is a very complex process, and we are greatly simplifying it by saying that electrons are transferred from the NADH and FADH to protein complexes and electron carriers. The electrons are used to generate a proton gradient, as protons are pumped across to the intermembrane space. All these protons being pumped out into this intermembrane space generates an electrical and chemical gradient. The thing is, if you remember from our cell transport video, ions, like the H+, don't easily travel across membranes directly without something to travel through. The protons can travel through an amazing enzyme called ATP synthase. If I could be any enzyme, I'd be ATP synthase because it has the ability to make ATP by adding a phosphate to ADP. ADP is a precursor to ATP. ADP has 2 phosphates, but if it obtains a 3rd phosphate, it becomes...
Okay, let's write down a few things here. All right, the first thing that we need to write down is that the electron transport chain occurs in the mitochondria. It is aerobic, it requires oxygen, and that proteins are pumped against the gradient into the inner membrane of the mitochondria through ATP synthase. And that there's one other thing here, and I'm going to put it in a box, and that's ADP is, it has 2 phosphates instead of 3. If it gets a 3rd phosphate, then it's adenosine triphosphate, but this is adenosine diphosphate because it has 2 phosphates instead of 3.
ATP by adding a phosphate to ADP. ADP is a precursor to ATP. ADP has 2 phosphates, but if it obtains a 3rd phosphate, it becomes ATP. So, in chemiosmosis, the protons travel down their electrochemical gradient through a portion of the ATP synthase, powering it to make ATP, the ultimate goal. Oxygen is the final acceptor of the electrons. When oxygen combines with 2 hydrogens, you get H2O, water. If you remember from our equation, water is a listed product. Now, the electron transport chain in chemiosmosis step makes a lot more ATP compared to the other 2 previous steps. How much? So I've noticed in my years of teaching.
Okay, so I wrote down that in this part, ADP receives another phosphate to become ATP again, and it releases water in that process. So the electron transport chain is the part of the process where the ADP that has had a phosphate bond broken to release energy for us to use is then become ADP and so it only has 2 phosphates. And when it goes through the electron transport chain, it receives the additional phosphate group and becomes ATP again so that the cycle can continue.
teaching and the assortment of textbooks I've collected over the years, there are some different numbers in cellular respiration charts. In fact, you can even see them change a bit in different editions of the same book. And it's made me want to emphasize that it's important to not just memorize a number, because it really is more of a range. I want to focus less on the number of ATP made per glucose molecule because it depends on a lot of variables. One variable is the gradient we were talking about, how many protons were pumped across the mitochondrial membrane. You can see more variables discussed in the factual references, and those references have estimates ranging from 26 to 34 molecules of ATP per glucose molecule in the electron transport chain and chemiosmosis step alone. Then, if you add the other two steps, Krebs, otherwise known as the citric acid cycle, and glycolysis, you could estimate a range of anywhere between 30 to 38 net ATP molecules total per molecule of glucose. Again, to emphasize a range. Now, this was just one way of creating ATP. But like we had said at the beginning, all cells have to make ATP, but the way they do it can differ. If there's...
Okay, so I just wrote the last note here, which is that there can be from 30 to 38 net ATP molecules produced from one glucose molecule.
No oxygen available, some cells have the ability to perform a process known as fermentation. It's not as efficient, but it can still make ATP when there isn't oxygen. We have a video about that. We can't emphasize enough how important the process of making ATP is for cells. For example, cyanide, which is found in some rat poisons, can block a step in the electron transport chain, which would block ATP production. A poison that prevents ATP from being produced can be deadly. With the important role that mitochondria have in ATP production, there is also a demand for increased research on mitochondrial diseases. We are confident that the understanding of how to treat these diseases will continue to improve as more people like you ask questions. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so let me point this out. So 30 to 38 ATP molecules from cellular respiration. And then we also have another way to do it, fermentation, that we can use. Came from one glucose molecule. And where do we get the glucose molecule? We got the glucose molecule from photosynthesis. So photosynthesis makes the glucose molecule, and then cellular respiration uses the glucose molecule to make ATP so that we have energy. Energy to move our muscles, energy for our body to beat our heart, blink our eyes, for cells to move. Anything that requires energy comes from the glucose molecule producing ATP. So our last video today is fermentation. And here it is. Let me make sure there's not a new one. Okay.
Hi there, we are the two amoebas behind our YouTube channel, The Amoeba Sisters.
goodness.
I'm Pinky, a former high school biology teacher, and this is my sister, Petunia.
Well, that did not work.
Melio's business payment solution is the simplest way to pay suppliers and contractors. Why? Because you can pay any
When I was a kid, swimming was one of my favorite things to do. I was on the swim team starting when I was 4 years old and then on and off throughout grade school. Although I wasn't especially fast. I just loved the water. I used to dream that there was a way I could be like a fish. When I was little, I had a misconception that fish didn't need oxygen. Later on, I learned that, no, most fish have gills that allow them to extract the oxygen, which they need, from the water. So then I just thought it'd be really cool if I had gills. But alas, no gills for Pinky. Oxygen is a really big deal. So many organisms, from fish to plants to humans, need oxygen. And yes, even though plants make oxygen in photosynthesis, they still perform cellular respiration, and therefore plants still need oxygen themselves. There's often a misconception that plants don't need oxygen. That's just not true. So why do these organisms need oxygen? It's similar to why you need oxygen. If you've ever wondered why you need to breathe, which is done by the respiratory system in your body, zoom into the cell level. Cells in your body use the oxygen you inhale to perform cellular respiration. The formula here requires inputs, otherwise known as reactants, to make ATP. And oxygen is one of those reactants in the overall equation that is needed to break glucose down in forming ATP. Why ATP? ATP stands for adenosine triphosphate, and it's action-packed with 3 phosphates. It has the ability to power many cellular processes. Typically, it's coupled to other things that it may be powering. Now, after losing the phosphate, the molecule is ADP, adenosine diphosphate, because it has 2 phosphates. In cellular respiration, though, there are enzymes that can add another phosphate to it and convert it back into ATP again. This particular formula of cellular respiration is aerobic, meaning overall it requires oxygen. It's pretty complex, and we have a video breaking down the steps. But that's not what this video is about. This video is about what happens when there is no oxygen, because cells still need to make their ATP. So what kind of cells can handle the no oxygen thing? Well, many types of bacteria can, many types of archaea, yeast, which is a fungus that could be helpful, like making your bread rise. Your muscle cells can, for a while anyway, and these are just a few examples. Now, these organisms handle the lack of...
Okay, so I wrote down, this is a process, let me do this too, process to make ATP without oxygen. Okay, it's a process to make ATP without oxygen. And I put the list of examples of cells that can do this.
Of oxygen in different ways. Some organisms, such as some types of bacteria or archaea, can do anaerobic respiration. They can continue to perform glycolysis, Krebs, and the electron transport chain, just like aerobic cellular respiration. But since there is no oxygen to be that final electron acceptor at the end of the electron transport chain, they use something else, sulfate, for example. These organisms are specifically adapted to be able to use a different electron acceptor in this anaerobic respiration. Another option is the organism may just stick with doing glycolysis, which doesn't require oxygen, and then the addition of some way to get their NAD plus back. We'll talk about what that means in a minute. This process is called fermentation, and that's what we're going to focus on. Fermentation is a way to be able to handle the little to no oxygen issue. It allows for glycolysis to happen and for glycolysis to keep going. That means making ATP where there is no oxygen. And while you won't make as much ATP in this process as you would aerobic cellular respiration, you can't be too picky when oxygen isn't around. Okay, so recall what glycolysis is from our cellular respiration video. In glycolysis, you take glucose, a sugar, and it gets converted into pyruvate. This takes a little ATP cost to actually start it up, but overall, per glucose, you make 2 net ATP molecules, and you also produce 2 NADH. Recall that NADH is a coenzyme and an electron carrier. We also need to mention that NADH didn't just poof appear as a product. No, because NAD plus actually was reduced to NADH when it gained electrons. And if the words reduced and oxidized are confusing, you can remember the famous Leo Gurr mnemonic. Lose electrons oxidized, gained electrons reduced. So,
okay, so lose electrons oxidized, gain electrons reduced. Okay. So I'll write that down for you.
NADH to NAD plus is oxidation because it loses electrons, and NAD plus to NADH is reduction because it gains electrons. Now, NADH, the electron carrier, would normally be delivering the electrons gained to the electron transport chain, if this was aerobic cellular respiration. Once losing their electrons, NADH would be oxidized into NAD plus and be ready to be involved all over again in glycolysis. But there's no electron transport chain in this fermentation process, so we've got to regenerate the NAD plus somehow. NAD plus is needed here, after all, for glycolysis to continue. Fermentation, therefore, adds another little step to the end of glycolysis, a step to help regenerate NAD plus. This happens because fermentation allows NADH to give its electrons to an electron acceptor, which in the two fermentation examples we are going to give will either be a derivative of pyruvate or pyruvate itself. So here we go with two types of fermentation, which both result in different products from pyruvate. Alcoholic fermentation is done by some types of yeast. So first glycolysis happens, which yields 2 net ATP, 2 pyruvate, and 2 NADH. Now we need this step to regenerate the NAD+, so we can keep doing glycolysis. The 2-pyruvate is used, which will ultimately produce carbon dioxide and 2-ethanol, alcohol. But the derivative of pyruvate shown here, acetaldehyde, can act as an electron acceptor in this process, so the 2-NADH can be oxidized to 2-NAD+, so that glycolysis can start all over. Since alcohol is a waste product in this process, this is why many yeast are used to produce alcoholic products. Yeast also can do alcoholic fermentation in making bread, and the carbon dioxide product we mentioned is involved with helping the bread rise. The tiny amount of alcohol produced in the short fermenting time of bread will evaporate in the baking process. Lactic acid fermentation is done by cells such as your muscle cells, for example. Well, your muscle cells can be electrons gained to the electron transport adds another little step to the end of glycolysis. In the two fermentation examples we are going, first glycolysis happens, which yields.
Okay. So let's go ahead and write this down, and we're going to circle where the problem is. So we have glucose that is converted an NAD plus into NADH, two of those, and we have ADP that is produced 2 ATP, and then we have 2 pyruvates. Okay, now the problem is right here. I'll circle where the problem is. Without oxygen, this NADH plus does not have anyone with which to receive the hydrogen from, because the hydrogen would usually come from the H2O where the oxygen has connected. Okay, and so
2 net ATP, 2 pyruvate, and 2 NADH. Now we need this step to regenerate the NAD+, so we can keep doing glycolysis. The 2-pyruvate is used, which will ultimately produce carbon dioxide and 2-ethanol, alcohol. But the
okay, so we have giving off CO2, and it's producing acetaldehyde, which is an alcohol.
The derivative of pyruvate shown here, acetaldehyde, can act as an electron acceptor in this process, so the 2-NADH can be oxidized to 2-NAD+, so that glycolysis can start all over. Since alcohol is a waste product in this process, this is why many yeast are used to produce alcoholic products. Yeast also can do alcoholic fermentation in making bread, and the carbon dioxide product we mentioned is involved with helping the bread rise. The tiny amount of alcohol produced in the short fermenting time of bread will evaporate in the baking process. Lactic acid fermentation is done by cells such as your muscle cells, for example. While your muscle cells can do aerobic cellular respiration, they can shift to lactic acid fermentation if they experience an oxygen debt. This could happen if you're working out very intensely, where your blood is unable to deliver a sufficient amount of oxygen to them for their demand. Just like alcoholic fermentation, we start with glycolysis that yields the 2 net ATP, 2 pyruvate, and 2 NADH. But now we need the step to regenerate the NAD+, and this step is a bit different from alcoholic fermentation. The 2-pyruvate on the reactant side will ultimately yield 2-lactate. The pyruvate can act as an electron acceptor, allowing NADH to be oxidized to NAD+, so that glycolysis can start all over. By the way, this lactate product, or specifically its other form, lactic acid, has often been blamed for the muscle soreness that occurs after intense exercise. In many of my teaching years, this was often discussed, but there's some recent research that may contradict that. Science is so cool. Check out our further reading suggestions and our video details to learn more.
Okay, so in lactic acid fermentation, which occurs in the muscles, you get 2 pyruvates, which convert into 2 lactate, and in that process, you have the NADH, which turns into NAD+. Okay, so let's recap what we've learned today, because this is a lot. And let me give you some suggestions on how to try to memorize all of this. So today we've learned 3 things. We've learned photosynthesis, we learned cellular respiration, and fermentation. Okay, we've learned those 3 things. What is the goal of photosynthesis? It's to make glucose. What is the goal of cellular respiration? It's to make ATP, which we need for energy to do for our cells to be able to do things. And then fermentation is an extra step, a different step in the cycle of producing ATP when there's a lack of oxygen. Now, if you'll open your book, let me turn this off so that it's bigger here. If I knew how. Oh, stop share. Okay, so if you get out your book, let me walk you through some helpful diagrams in your book. Turn to page 180. On page 180, you'll see the 3 things listed in the middle that your ATP is used for. The mechanical work, like the movement of chromosomes, the contraction of muscles, the beating of the cilia or flagella on the cell movement, transport work to move things within the cell, and chemical work like making larger molecules from smaller ones. So those diagrams on page 180 are just showing you what is ATP used for, why do we need it? And they're giving you examples of ways cells use the ATP. On page 181, they're showing you exactly what the video showed you, that the reactants in photosynthesis become the products in cellular respiration. On page 182, you'll see that it shows you ATP at the top, where it has the adenine, the base, the ribose, and then the 3 phosphate groups, but that when you break off a phosphate bond, you release that energy by breaking that bond, then you get adenosine diphosphate, ATP, which is at the bottom, where you still have your adenine, your ribose, but you only have 2 phosphate groups bonded to it instead of 3. Okay. All right, on page 189, this is showing you photosynthesis. You see the light entering, and you see the electrons moving, which have to do with your NADP and your NADPH. You see your ATP produced from your ADP at the end with the Kelvin cycle there going from the NADPH up to the Kelvin cycle. So a little bit different than the way we drew it. So this might be more complicated than the way we drew it in our notes. So if this is not making sense, just use the one that we drew in our notes today. Let me see if there's, and again, same thing on page 192. Let's see here. Yes, again, a little bit more confusing than what we drew today, but feel free to read through that and compare it to what we drew. Then you have the Calvin cycle on page 193, which we talked about. And you see the chemical reaction that we wrote down there. So that's exactly like in our notes. Again, the Calvin cycle, again on page 194 in more detail than what we wrote down. And then on page 200 is the Krebs cycle, which we wrote down. And on page 202 is the electron transport chain. And then on page 205 is fermentation. And that is all. So I hope that the notes that we wrote together today are a little bit more helpful than the diagrams in your book. But make a list of questions that you have for Wednesday. I have put up the new work that needs to be done. Of course, remember, don't forget your Module 4 test. There is an Amoeba Sister video on the parts of the cell that might be good if you're struggling with getting ready for that test on the parts of the cell, where they're located and what they do and what their name is. That's the entire test. So be sure you're studying for that. And then I did put up your on your own, your reading and your flashcard schedule on Jupiter Ed as well. I look forward to seeing y'all on Wednesday. We'll do a lab on chromatography on Wednesday, and we'll talk more about the things we discussed today. So I'll see you then.